All right, good afternoon, everybody. We're at 12:02 now, and I see a good number of people that are on at this point, so we're gonna go ahead and get started. So thanks for joining us today. This is the last of our four Northeast IPM question series. Used to be the stupid questions, but the more we do this, the more advanced we're getting. But again, the purpose here is to kind of learn about new topics together. There aren't any growers on the call, so very relaxed, informal. Just to learn about a couple different topics that we've kind of wanted to go a little bit deeper dive on a couple of different areas. So from the feedback we got from you all, the topic of ambrosia beetles came up near the top. I think if we think back to the meeting we had back in Massachusetts in October, we had a pretty good conversation about them then. So I think that really kind of launched us wanting to have this talk today. So luckily for us, we're joined by Dr. Kelsey Tobin from Cornell Agritech. She's a postdoc in Dr. Monique Rivera's lab, and I think Monique's hiding in the background too today, so she'll be here as well. Before joining the Rivera lab, Kelsey got her PhD from Purdue in the forestry department. And I know she's been working on ambrosia beetles while she was in Indiana, and then continues to do so here at Cornell. So very excited to hear about some of the work that she's been doing, looking at different traps and different repellents. And also she has the bad news of telling us about a new species of ambrosia beetle she found this past season. So without further ado, I think I'm going to go ahead and turn things over to you, Kelsey. Thanks for joining us today.
Sounds good. Thank you. And let me share the screen, get it loaded up here. Let's see. Let's see it. It's not working. We tested this out before and it worked, I promise. Okay.
Looks good.
Hopefully you can see the screen. Okay, so I am Kelsey Tobin, and yeah, I'm currently located in Geneva, New York, working primarily on ambrosia beetle pests in our apple orchards. So I'm going to provide a little bit of an outline of this talk today. I'm going to probably only take like 30-ish minutes to talk so that we have plenty of time to discuss or answer any questions you may have towards the end. But I'm going to go over the ambrosia beetle biology and why we have such issues with their management just because of their biology. It's pretty different from the other pests we have in apple. And what our current strategies are, and things that we're working on to try to improve those management strategies. We also have looked at different biotic and abiotic stressors and how those influence beetle attacks. And this past season, we tested a push-pull management system to try to keep the beetles out of entering the orchard rows from the woody edges surrounding the orchards. And then kind of what we're continuing to work on based on the results that we've seen from these projects. So hopefully all of you are aware of the black stem borer, Xylosandrus germanus. This has been our resident ambrosia beetle in apple since it was first detected in 2013. It's got a really broad host range, so it's not particularly just a pest of apple. It's got over 200 hosts, and so that's why we think these unmanaged woodlots that surround our forest edge are a really great habitat for them to reproduce and overwinter, and then they can move into our orchards from there. It was first detected in the United States in the early 1940s, but wasn't declared an apple pest until 2013. And they've caused some economic damage to the trees, but a lot of times that's sort of a secondary problem that comes in after late freeze or fire blight or flooding, some sort of other primary stressor that draws the beetles in. Black stem borer typically in our area has 2 peak flight periods in late May and then again 1 month later in mid to late June. And so the current recommendations for this species are to target pyrethroid sprays around those peak flight periods. But we've seen over the couple years, depending on if we have a really severe or mild winter, that those peak flights can fluctuate even by just a week or two. And so trying to time those preventative sprays can be really challenging when we have a lot of environmental variability that's influencing when the beetles fly. This picture here in the middle shows the females on top and the males on the bottom. The species is haplodiploid, so only the females are flying. The males never leave the host tree. So our primary management is for those adult females. Here's just some examples of what their tunneling looks like inside of the trees. You would never really see this unless you crack open an infested tree. And although these beetles are really small, the extensive tunneling caused by multiple infestations or really large infestations of these beetles can girdle your trees, especially young trees that are not very large in diameter, or branches that are also particularly small. And in our high density orchards where our trees are never really putting on much size, they're going to be a little bit more susceptible to some of this more extensive damage. So these beetles bore into the wood. Here are a couple examples of what the boring holes look like. Occasionally you'll see some sap dripping out from around these holes, or you'll see compacted frass, or like wood shavings essentially that come out. They kind of look like noodles. I don't have a great photo to show you. Oftentimes they don't bore specifically on these smooth bark areas. They'll bore more likely in these old like wounds or pruning cuts, which makes it a lot harder to actually even see the holes in general because the bark can be a little bit more rough or in some sort of crevice. Once the beetles bore into the tree, they line the galleries that they've excavated with a symbiotic fungus. For black stem borer, it's called Ambrosia grassmanniae. And the beetles don't feed on the host tissue of the tree, but they feed on this fungus instead. And with extensive damage, the trees can die after a couple years. And yeah, some growers have to tear up whole entire plots or specific trees in their area. So some of the current management recommendations are just to keep your trees healthy, but most of you probably know that's not always in our control, especially some of the environmental factors like freezing and flooding. And we get kind of different outbreaks of disease from year to year. So it's really difficult to say maintain tree health to avoid ambrosia beetle damage. The following recommendation is just these preventative insecticidal sprays, typically using pyrethroids. And it's just recommended to apply these throughout the growing season. Sometimes it's in the grower's regimen in terms of spraying for other pests as well, but these compounds can affect the natural enemy populations for ambrosia beetles, but for some of our other pests of interest as well, like woolly apple aphid. So if you're constantly spraying these insecticide sprays for beetle prevention specifically, you're going to be targeting a lot of non-target beneficials in the process. So to break it down a little bit further about the beetle management, we have one female beetle that can bore into a tree, and then she'll lay the eggs inside the tree, and those eggs develop into larvae and pupae and adults. Now, in the first stage of ambrosia beetles in the early spring, when they lay that first round of eggs, they all are going to be males because they have a haplodiploid biology. And so then the one female mates with all of those males, and then all of those offspring become females. So it takes one single female to create an infestation. And in that second round, you can get anywhere, depending on the size of the tree, anywhere from like 10 to 60 females from that one individual female. So it can really start to grow exponentially once you have a single female that's able to establish. So in terms of management in these 3 life cycles, these all occur inside the tree, so they're protected from any pyrethroids that you're going to be spraying. There's been some early work to look at fungicides that specifically target the fungus that the beetles feed on, or relying on predators and natural enemies, pretty much other larger wood-boring species that can feed on the larvae and pupae of ambrosia beetles. but the natural control that you're going to get from these populations is pretty minimal. So with those preventative pyrethroid sprays or insecticidal nettings to use on adults are the two management practices. The insecticidal nettings are going to be really hard to deploy at a large scale, especially in the high density orchards. So really, other than pyrethroid sprays, we're like, what should we do? These beetles also don't produce a species-specific pheromone, so we have to rely on host chemicals from our apple trees that are specifically luring those beetles into the host. And since I mentioned that these beetles prefer stress trees, one of the key signals that they're cueing in on from those stress trees is ethanol. Under any amount of stress, a tree will release ethanol as a byproduct of anaerobic respiration. And so we can use this ethanol on our different styles of baited traps to lure those ambrosia beetles into the traps rather than our trees. And we recently did a study that compared a couple of the commercially available trap designs that are available. And we found that the sticky cards, they're just about probably 8 inches long, and they have this sticky adhesive glue on both sides. That trap design was the best at capturing and retaining the most beetles, but also a broader diversity of pest species across Eastern United States orchards. We did this here in New York and also in Georgia. So they have different species composition down there, but it was able to capture all of the pest species that we were looking at. These other trap designs still capture a lot of beetles, but they might require a little bit more servicing than the sticky cards. And so going off of this ethanol cue that these beetles are interested in, we know that stress trees are producing ethanol, but we don't necessarily know how much ethanol is coming off of these stress trees. And so we wanted to look at specifically 2 stressors that we see commonly in apple and observe the ethanol emission as well as beetle attacks on fire blight trees as well as flooded trees. We did this in potted trees as we weren't able to flood a field. And so we did this in potted apple trees. And we found the most beetle attacks on our flood-induced trees. They were flooded for solidly for 2 weeks, which is probably not super what we would see in the field of flooding conditions, but we kept them flooded fully for 2 weeks. And by the end of the 2 weeks, they were pretty much dead. But they had the highest number of beetle attacks. And then our fire blight trees also had significantly more beetle attacks than our untreated control trees. And so we can pretty confidently say that the beetles are more attracted to these stress trees than our healthy trees. Although our trees, even though they might appear visually healthy, they might still be under some sort of stress and emitting ethanol. So it's not always like a visual, oh, this tree has fire blight, oh, this tree's flooded. There could be some other underlying conditions going on that are causing the trees to produce ethanol. When we tried to look at the amount of ethanol being emitted from these stress conditions, the active range of the fire blight that was in sort of the necrotic tissue that you typically see in the wood of fire blight, they had a really high ethanol emission rate. On those same trees outside of the active fire blight range, we took ethanol concentrations and found that there was still some ethanol being produced, but at a significantly lower amount than the active range. We unfortunately right now don't have the data for the flooding tissue. The trees ended up being pretty much dead by the end of the 2 weeks, and so the tissue was unable to be analyzed, but we're trying to find a sweet spot where we can flood the trees without killing them and then take the ethanol concentration. And in our healthy trees in this scenario, there was no ethanol being emitted from the healthy trees. And so if you see boring activity on healthy trees, there might be something underlying there that is not necessarily a visual representation. And so this was all done with black stem borer, Xylosandra germanis. And when I got to Cornell last year, in the spring, we found a second species of Ambrosia beetle that is attacking apple trees. This species is called Anisandrus maisch. We don't currently have a common name for this species. We are working on it, and we don't expect growers to remember the scientific name. And this beetle is attacking apples as well. We found it in really high infestation levels in particularly young orchards that did have a fire blight outbreak this past spring. And we've also noticed some differences in the biology between this species and black stem borer. I mentioned that black stem borer has those two peak flights in May and June. And for Anisandrus maisch, we see it starting to fly here in New York around mid-June, and we get consistent trap captures from mid-June through August. So we don't have those peak weeks where you're going to see a lot more beetle activity. It's a longer and less pronounced flight with consistent weekly captures occurring through the second half of the growing season. So Xylosandra germanis, there's been quite a bit of work done on this species, particularly in apple, since it's been a pest for over 10 years. We know it's attracted to ethanol, and Art Agnello did some work with this compound verbenone that's been shown to be a repellent for many wood-boring species, particularly ambrosia beetles, and also some bark beetles that are very closely related to ambrosia beetles. It's actually an anti-aggregation pheromone in bark beetles, which signals to other species and the same species to not attack the same tree. We know that ambrosia beetles don't produce this compound themselves, but they are repelled by this species, as it might indicate a host is not suitable. While I was at Purdue, I actually studied Anisandrus maisch in hardwood trees and did some work that confirmed that this species is also strongly attracted to ethanol and repelled by this compound verbenone. There's a third ambrosia beetle species that here in New York we don't have. I'm interested to hear if anyone on this call has experienced this species, Xylosandrus crassiesculus. It's also called the granulate ambrosia beetle. It's a lot more common in the southern United States, but we recently did a study with Pennsylvania, and they had a high abundance of this species as well. And lucky for us, this species has the same response to these semiochemicals, a strong attraction to ethanol, and a strong repellent by verbenone. And so since we see this consistent overlap across the response to these chemicals, we wanted to test them in what we consider a push-pull management design. And the ultimate idea for this was to deter the ambrosia beetles from our high-value apple trees by pushing them away with a deterrent and pulling them into these attractive baited traps that had ethanol on them. I mentioned that most of these ambrosia beetle species have hundreds of hosts that they're able to attack. And so a lot of our population within the orchards is actually coming from those unmanaged woodlots. Once the temperatures warm up in the spring, the beetles will emerge from the hosts in the forested edge and migrate into the orchards, where they will start to dig their tunnels into the trees. And so our idea in this design was that we wanted to intercept the beetles along this wooded edge by using our ethanol baited traps. And this project was done in collaboration with folks in Chris Ranger's lab in Ohio at the USDA ARS and Greg Krawczyk at Penn State. So we deployed this in commercial orchards across the 3 states. And at each state, we had replicates of this push-pull design that included the ethanol baited traps along the edge with our push of the verbenone, and then just the verbenone alone without any ethanol along the edge, and then just our ethanol baited traps. These blue triangles within the orchard rows, we used a really, really low release ethanol on those sticky card traps. Mostly because just an unbaited sticky card isn't going to capture many ambrosia beetles. We wanted to have that ethanol signal in there as like a similar to a tree being stressed, but so that we would actually be able to see if those beetles are penetrating the repellent wall and getting into those orchard rows. We did a little bit of a cost analysis for what we specifically designed in our study this past summer. In each of our subplots, we used 100 grams of verbenone, which is still in the process of deciding if that's maybe too much. We wanted to really start out strong and then try to dose it back as we could. The nice thing about these verbenone sachets is that they last for about 8 weeks. So you would need reapplication throughout the growing season, but 8 weeks is a pretty solid amount of coverage. And that coincides with some of our ethanol lures that we also replace every 8 weeks. And so you could go out and replace these at the same time. We were collecting our sticky traps every week for our purposes, but just for monitoring purposes, I think you could get away with changing them every other week or maybe even less frequently than that. So we did deploy this in a commercial orchard along the edge. We set it up really early last year in the spring because we had such a mild winter here in New York. We were worried about an earlier flight time for specifically black stem borer. But I think we set it up a little bit too early. We didn't start to catch beetles until May. So I think we set this up the first week of April. So they were out there and just not capturing anything. But here's the verbenone sachets that we attached to our trellis wires. And then in this last panel is one of the sticky cards that we also attached to the trellis wires. And this is the ethanol, low dose ethanol lure. So I'm going to go state by state with our results that we found. The trends are pretty consistent across states. So I just want to point out that we do see similar trends. For starters, we captured black stem borer in the highest abundance here in New York with about 1,100 total between the high and low ethanol baited traps, about 500 Anisandrus maisch, and we did capture 59 of that third species, Xylosandrus crassiesculus. But in our case, we only ever captured that species on those orchard edge rows. We never caught any inside of the orchard. So we don't see this species as a pest currently in New York State. We do have it, but it's in really low numbers. And so we didn't analyze it in the New York data. So to orient you to these figures, the black panels are going to be those border edge high dose ethanol traps. And the gray bars are the low dose ethanol traps that are in the orchard rows. So we pretty consistently captured similar numbers on the high dose ethanol traps that were right adjacent to the edge, about 10 beetles per week. But we did see a significant reduction when we used that verbenone component in the push alone, which was just the verbenone, and the push pull, which was the verbenone paired with the high dose ethanol lure. We saw very, very few beetles able to enter those orchard rows. And this was over a 10-week period. For Anisandrus maisch, we saw a similar trend. We caught less of this species overall, but in both cases of the push and push-pull where that verbenone was present, we didn't have any beetles entering the orchard rows. So we can say for New York, the verbenone component significantly reduced beetle capture in the orchard rows. In Ohio, they have higher beetle numbers than we have in New York, with about 3,000 black stem borer, 1,500 Anisandrus maisch, and about 600 Xylosandrus crassiesculus. But the trend continues that we still see a reduction in beetle capture when we have the push component. And that tracks for black stem borer, Xylosandrus crassiesculus and Anisandrus maisch. Since they do have slightly higher beetle pressure, we did see on average 2 to 3 beetles entering those orchard rows each week in the verbenone treated blocks, but it was a reduction from just our ethanol traps alone. And lastly, in Pennsylvania, the species composition changes quite a bit, with them having upwards of 6,000 Xylosandrus crassiesculus, only about 150 Anisandrus maisch, and less than 600 Xylosandrus germanus. We didn't see any differences in Xylosandrus germanus with that push component, but we had very few beetles entering the orchard rows in general over that 10 weeks. With Xylosandrus crassiesculus, we saw a reduction with the verbenone alone, but the beetles were further reduced when you combined the ethanol-baited traps along the edge and the verbenone in the first and third orchard row. And we barely caught any Anisandrus maisch in this experiment, so we weren't able to do like a robust statistical analysis, but we didn't see any Anisandrus maisch in either of the plots that had the verbenone component, and a few in just the ethanol alone. So the only time we saw the additive effect of having the ethanol-baited traps paired with the verbenone component was with Xylosandrus crassiesculus in Pennsylvania. But we think this is really strongly population dependent. If you have an orchard where you have really, really high abundances of any of these species, we think that having that ethanol-baited trap will help further reduce beetle activity in the orchard. But if you have mild to low populations of beetles, that just the verbenone component alone can reduce beetle entry into those orchards. So we're still working on identifying this verbenone component and the best way to deploy it. We do see the reduction. So we know that the compound is affecting the beetle movement and migration into the orchard. It is population dependent, so depending on your pest pressure, you might want to incorporate those ethanol traps. But there are different formulations of this compound that are available. Here's a component or a product called splat verbenone, and it's a waxy sort of putty that you can apply directly to a tree. This product requires state-by-state regulations dependent on where you are. And last year we did not have a permit in New York State to test this product. So going forward, we have one for this year and are going to test this product in the field and try to identify a better release rate and hone in on how much of this product you would need to have that beetle reduction. One nice part of this product is that it is biodegradable and food safe. And with the verbenone pouches, you have plastic waste and you have to reapply them, taking them down. There's some waste that comes with that. This product is biodegradable. We're not sure about the reapplication yet. That's something else we're looking into this summer, is if we can go the whole season with just one application. And they also have some technologies. This can be applied with a attachment that goes on a tractor, or it can be applied with a drone. So a little bit more flexibility in terms of it doesn't have to necessarily be hand applied. So this year we're going to compare those pouches and also the wax matrix to see if anything is better or worse, if we can get any recommendations from this experiment. And we also want to pair these verbenone, whether it's the pouches or the splat, with these really stressed trees. So we're going to attempt again to flood trees without killing them and do some fire blight inoculations and apply the verbenone to those trees and then observe beetle attacks, since we know that the beetles are preferentially going for those stressors. And the last little experiment I want to talk to you guys about is some work that's being done by a PhD student in the lab, Sandra Lizarraga, and she's looking at the effect of time of day and temperature and kind of the circadian rhythm of these ambrosia beetles to better optimize trapping, as well as some of those preventative sprays that I mentioned early on. So she did an experiment last summer by trapping in different hour increments to try to identify any sort of temporal peaks of flight patterns throughout the day. And this was again deployed along that forest edge where we think most of our beetles are migrating in from. And her time groups were 8 to 10 a.m., 10 to 12, 12 to 2, 2 to 4, 4 to 6, and 6 to 8 throughout the day, collecting those traps every 2 hours. And at the same time, she was collecting environmental data on temperature and humidity using an iButton to look at also the effects of temperature on beetle capture. And all of this work was just done with black stem borer, so keep that in mind. And we found that the peak flight throughout the day for these beetles is after 2 p.m. We caught significantly more beetles between 4 and 8 p.m. And that was consistent across the 10 days that these beetles were captured. So this late afternoon and evening flight is maybe when they're more active. And that could be good to know when growers are doing these preventative insecticidal sprays, trying to optimize time of day with beetle activity so that you're targeting those flying beetles. She also compared this with the temperature ranges and found that 20 to 25 Celsius or 68 to 77 Fahrenheit is a preferred flight range for these beetles. And she took it one step further to correlate time of day and temperature to make sure there was no effect there. And the frequency of these temperature ranges throughout each time of day was pretty consistent across all of the time groups. And so we can pretty confidently say that it's not, oh, because it's warmer later in the day, the beetles are flying. We saw this 20 to 25 temperature range across all of our time groups. So after 2 p.m., especially if the conditions are 20 to 25 Celsius, we will expect to see black stem borer activity at its peak. And so helping to depict some of these temperature and environmental factors is going to help us identify optimal timing for these sprays as we continue to still use pyrethroids in our IPM programs for ambrosia beetle management, as well as combining it with some of these other trapping protocols and repellent compounds to more effectively manage overall beetle populations. We are going to do this trapping timing study again this year and include Anisandrus maisch to see if there's similarities in timing of flights or temperature preferences for this species. And ultimately, we want to use some of this data to develop a model to better predict beetle flight based on environmental factors and potentially, like, if we have a more mild winter, can we predict if the beetles are going to fly earlier in the season, later in the season, and how that might affect overall beetle populations from year to year when we have significant climatic variation. And that is going to wrap up what I have to present. I'm excited to hear any questions you guys have, or if you would just want to have a discussion about ambrosia beetles, I could talk about them all day. So I will turn it over to you all, I guess. Thanks for listening.
Right. Thank you very much, Kelsey. Really, really great work and interesting to see where the research has been heading on all of this. So with that, I would like to definitely open it on up. If people want to type it in the chat box, or if you want to unmute yourself, feel free to ask away any questions. We've got plenty of time, so feel free to fire away. And while
I'll ask
people, oh, go ahead.
Hey, Mike, thank you. That was a great presentation. Thank you so much. I just had a question. I'm curious. So on the cost associated with the verbenone and the lures as part of the push-pull project,
Yeah.
have you had any conversations with growers and gotten any feedback from them about how receptive they would be to that sort of thing? And just curious of what kind of input they're giving you on that. And I asked that mainly because um, we've been having this conversation here in New Hampshire with Jaime Pinheiro, who's in Massachusetts, looking at ways to better control plum curculio and apple maggot fly, and just cost around those practices is something we're pretty conscious of, and trying to think about creative ways to make that more appealing to growers, including really having frequent conversations with our partners at NRCS to see if that's something that they might include in some of their IPM offerings. So just curious to get your take on that and if you've had any of those conversations.
Yeah. I specifically have not had any direct conversations about the cost. Um, I think something that we're still trying to tease out is the dose that would be required to have this deterrent effect. Um, Based on the study we did last year with that 100 grams of verbenone, I think that was a little bit of overkill, but it's also really expensive to treat even a quarter acre. Um, and so yeah, we know the compound is deterrent. We're still trying to pinpoint the recommended amount, but the cost is definitely an important factor. We specifically strategically did that edge effect in hopes that instead of saying, oh, you have to treat your whole orchard with this expensive compound, um, trying to see if we can really deter them just by treating the edges. So that's where we're at. I don't know if Monique she can maybe comment more on the specifics in terms of costs and directly talking to growers about it. Maybe.
I think your answer was great. We haven't really spoken too much about grower cost yet because we haven't refined the method enough for a recommendation. But what we do provide is a start. I think when this problem is a problem, it's usually major. So having something that's not spraying additional pyrethroids, which we know can kill off natural enemies for other pests we're trying to keep in check, particularly woolly apple aphid. This is the best of what we have right now. We hoped in a couple years have some refined recommendations.
Just as a follow-up on Jeremy's question, I mean, I've seen the splat product, and I would imagine for growers that's going to be tough to, convince them to go out and put that on every so often. Is this verbenone, is this a product that you think could eventually be formulated into a sprayable material, or is it just the nature of the chemistry that's not going to be an option for us?
We have talked with the industry folks about having it be put in a more sprayable formulation, but if they did say that when you add water to the product, it really decreases its duration in the field. It does sound like that's something that they're actively, working on and hoping to maybe use adjuvants or something that can help the longevity of the product in the field. But the nice thing about this like waxy thing is that it's a slower release and it's going to last you for 12 to 16 weeks, I think is what they say is their average release rate. So yeah, I think if you do start to water it down, you're going to get less coverage. Well, maybe more coverage in terms of aerosol, but less duration, longevity of the product is my concern.
Okay, great. Thank you.
Yeah.
We do have from Lisa, she mentioned in the chat, I think just reaching back to your question to the group about what people are seeing. So she mentioned about, 2,600 Xylosandrus that they caught in hardwoods in Rhode Island. Most of them in the first 2 weeks of trapping in June. And they haven't checked in apples, but in that same trap only found 100 Xylosandrus. Lisa, did you want to comment anything else on that? Or if other folks want to also chime in about what numbers they're seeing around the region, I'm sure that would be of interest to the group too.
One thing we do see with the Crassiesculus is it has, similar to the Anisandrus maish, it kind of flies. It seems like for longer during the season, but June would be when you start to see it popping out.
Okay. I don't want to throw people onto the screen here, but I see Caitlin's on from New Jersey. It does sound like species do kind of shift the further south you get. Wondering, Caitlin, or what you might, if you're looking at these, and what kind of spread of different species you might be seeing down your way.
Yeah, so this season was our first time actually trapping for them. Moving into next season, we're gonna be working with the plant diagnostic lab at Rutgers to more closely identify what species we're seeing, but we have confirmed through like bringing in trees that have like had dieback and had the ambrosia beetle holes being seen. We brought them into the diagnostic lab and they found both black stem borer most commonly, but there's also granulate ambrosia beetle a little bit in North Jersey, but that tends to be skewed a little bit more towards South Jersey, the granulate ambrosia beetle. Yeah, we haven't found the new one that you're talking about yet, but I'm very curious to see if when we start identifying some of the species next season, if we see some of that as well. It would be really interesting.
So Caitlin, quick question. These are going to Richard Buckley. That's he's still running the diagnostic lab at Rutgers.
Yep, and Sabrina Turpak is there too. So she's doing most of the identification.
Yeah, I might reach out to them just because they might need additional training on the difference between these two species. They're really close.
Yeah, that would be great if you could do that.
Okay, will do.
Yeah, thank you so much.
All right, thanks, Caitlin. So we have a question from Glenn. Do growers in the southern US or anywhere target treatments against their ambrosia beetles? Do you know about the treatment in other regions?
I can talk about that a little bit. So I would say that the biggest threat caused by ambrosia beetles in the continental United States is really the threat of carrying the lethal fungal pathogen laurel wilt. So I worked in this system in Florida, in avocado, where that pathogen is spreading. So in avocado, again, this is a really high dollar cropping system, especially in southern Florida, where they grow really very diverse varieties. It's not like California or the West where it's mostly Hass. So essentially what they try to do is they try to improve their nutrient program, but in terms of species, they have, I would say, about 15 species, and they're not quite sure who the true vector is. So there are some applications there, but they don't target the insect in this case. They, in order to protect the tree, because it's acutely this fungal pathogen, they're using fungicides, which are usually trunk injected inside of the tree. So it's a totally different system. I don't think trunk injections are going to save us in this particular system. And I think that we have timed flight by season, where in Florida, they're flying literally all year long, and it's a bunch of different species, and there's a bunch of questions about promiscuous associations with the pathogen. But in terms of specific treatments in the southern US for ambrosia beetles, in, let's say, like tree fruit, I think they're in the same situation as us, where it's a problem, and generally speaking, pyrethroids are the recommended treatment for control.
Okay, thanks, Monique. I think a follow-up to that, Glenn was asking further, do you feel like the risk from either of these species right now is high enough to require targeted sprays and unstressed high-density plantings? And I see Vaughn commented on that too, so both of you are certainly more than welcome to chime in there about what you've been seeing around, certainly in Western New York.
I
think
haven't seen or heard of any reports of high attacks in unstressed plantings. It's typically what we saw was in ones that had already incurred some sort of stress. So I think currently the risk is maybe not, I would say not high enough. But if you're seeing attacks, either there's an underlying stressor that is already there, or I haven't seen any reports of attacks in unstressed.
So I can comment on that a little bit from our experiences. I would agree the best option by far is to reduce stresses. One of the things we've done in our region that has significantly helped is really push growers making sure that there's adequate drainage installed in orchards before they plant, and disease control and all that. That being said, young plantings and nursery plantings, and at least here in New York, get pushed so hard for growth that a lot of times there's stresses there that we may not necessarily recognize. And so the result of that is we have seen major tree losses in blocks and in nurseries that we thought were not stressed, and they probably were more than we thought. Sometimes to the tune of hundreds of thousands of dollars worth of damage. So yeah, we do everything we can to try and get growers to reduce stresses, but in those young plantings, we do still routinely spray. Not to say that we necessarily think it's always effective, but it's cheap insurance, unfortunately.
And so Vaughn, when they're spraying, are they doing those two pyrethroid applications?
Yeah, so we're doing pyrethroids. That is, really about the only thing that we think we have has any effectiveness. And the reality is, in those young plantings, of pyrethroids are also inexpensive control for other pests, and we do try to get away from pyrethroids somewhat once trees are mature. So we figure if we're going to push pyrethroids, do it in younger trees, and then try to shy away from them more later on when we really are more reliant on those beneficials. Not
Sure.
necessarily saying that's the best tactic, but that's what we've been doing.
Right, at least to get you by at this point. So when they're doing
Yeah.
those, it's full tree with the air blast cover the whole canopy.
Yeah, yeah,
Okay.
Vaughn,
I
are you
wanted-
in Western New York?
yes.
Okay, thank you.
Just to follow up on some of the things you said there, Vaughn. The question of drainage and I've been scouting for these in the Champlain Valley, and I haven't seen a lot of damage, and I've always kind of assumed it's because we generally are on kind of sandy loam soil, so we don't tend to have a lot of standing water and that kind of stressors. So this year there was one block where we did start to see some pretty severe damage, and it was young trees, and it was the one block in Peru that is planted on, moderately to somewhat poorly drained soil. So, um, with that in mind, I mean, it sounds like you're seeing similarly. It does seem to really, I mean, wet ground is a big issue for us. Do you feel like, so I mean, are you seeing it in fields that don't have any tile at all? Do you see it in some fields now where maybe the tile lines need to be reassessed because they're just not engineered to the point where they're able to move enough water? I mean, what's your kind of sense of that?
Yeah, so in our experience here, as more tile drainage has gone in, tree losses have declined. A, a lot of our soils here and a lot of our landforms here do result in situations where even with tile drainage, we still have areas of the orchards that will go underwater for periods of time. We try to reduce that as much as we can. There's some ground that, quite frankly, you could have a tile line every 5 feet and it probably wouldn't be enough. And quite frankly, that probably shouldn't be planted to orchard, but sometimes it gets done. What I would say is it doesn't just have to be excessive moisture stress. That's one of the things that we have seen in our region because of our soils here. But I do know that, drought stresses, disease stresses, all of that can also play a role. It just has not been generally the biggest factor here, although with the showing up of the maish species in this area, it was associated with a fire blight infection. So, I will say that, and no obvious stresses otherwise in that area.
Okay, great. Thanks, Vaughn. I just kind of moving on that similar vein, a few Dave and Glenn both kind of asked, and I think we've kind of just been walking through this. Um, and Kelsey, if you want to talk about what you've been seeing as well, on the other side of it, drought stress being involved in making trees attractive. Um, so we're seeing both of those, and like Vaughn just said, many other factors that are stressing trees out. Do you have roughly out of the blocks you've seen, not to get too nitty gritty, but what percentages are you seeing it kind of falling into where you're kind of getting at the primary cause here?
Well, I've only been in the Apple system for 1 year, so maybe take it with a grain of salt. But from the calls we received last year, it seemed like most of the blocks had fire blight or potentially like sudden apple decline. But I was never in a flooded or droughted orchard, at least not actively flooded or droughted, whether it had been earlier or later in the season. But I think last year fire blight was the most co-occurring stress with beetles that I personally saw. We did not, when we did the potted trees, we did not do drought stress as a treatment. But from other work that's been done in like other ornamental trees, drought stress is also does bring beetles in, but it does seem like flood is more attractive, I think, releasing more ethanol than a drought stress tree. And that's in part because a drought stress tree can still undergo respiration normally, where a flooded tree, when the roots are like fully underwater, they have to go into an anaerobic process. And that's where the ethanol is really starting to come out of those trees.
Okay, thanks.
And
When you did
another,
go ahead,
Oh.
Vaughn.
I was just going to say another thing that it doesn't always have to be environmental stresses or disease stresses. One of the things that we have seen occasionally here as well is attacks after trimming in young orchards or after budding activities in nurseries. That has happened occasionally too. So again, may have also been other stresses, but those types of activities can also stress trees and get them producing ethanol that will draw them in.
Okay, thanks, Vaughn.
Yeah.
So it sounds like really any type
Any
of
stress
yeah.
is bad.
Just don't stress the trees out at all. All right.
Yeah, which is not easy to do.
Right, right. I do want to go back. Jeremy had the question, kind of getting into the nitty-gritty scouting and monitoring. What are your recommended protocols between the different traps that you've seen? What are your kind of recommendations there? What do you for the us extension agents on the call that want to keep a closer eye on these things? What do you recommend? And I'll throw in there too, do we have any sense of thresholds for what a high risk level might be?
I would say those ethanol baited traps are the best for monitoring. Any trap style that's baited with ethanol will capture beetles. We did find that those sticky cards capture more overall, but any of the commercially available traps, or you can make your own with like basically cutting panels out of a 2-liter soda bottle, hanging the ethanol from the inside, and then capturing them in a cup. I can send some sort of like little info sheet on how to make those traps if that's of interest to anyone. Those sticky cards are commercially available, but I think they're actually recommended for brown marmorated stink bug, not ambrosia beetles. So if you want to go looking for them, they're stink bug traps. And yeah, the higher ethanol release that you can get from a lure that you put on those traps, the more beetles will come. There are commercially available ones, or you could just buy, I think you can probably just maybe buy ethanol I think, well, you can buy rubbing alcohol, but that's different. I think you can just buy ethanol from the store, maybe.
You could use liquor, like it's the
Yeah,
same.
anything with ethanol in it. If you have an area along a woody edge, that will give you a good idea. If you have a younger block that the trees are likely more stressed, or an area that does flood every year or every season, I would prioritize putting the traps in those areas that you've identified as higher stress. But along the woody edge, you'll definitely catch beetles once they start flying. Threshold, I think the current recommendation is once you catch them on the trap, start spraying, is my knowledge. Because once those first ones start to pop up in the early spring, it's warm enough for them to start flying. And those are sort of the like pinpoints that we have right now for targeting beetle flight.
Okay, thanks. Follow up on that. Time between like first catch and when you really start to see peak, is it how many weeks would you say, or is it pretty
It's
quick, or
pretty
depends on the year?
quick. Once the days are like consistently, I would say above 55 Fahrenheit, you'll start to capture black stem borer. But usually, yeah, you start to see that early May, and within a week you would have enough to be spraying.
Okay, wow, okay, good to know. Dave asks, are most initial infections found within a foot or two of the soil line, or can they be higher in the tree? We're usually looking on the trunk there.
We have seen them from high to low.
Say though that it's important to note that the higher ones tend to be in younger trees.
Yeah, the larger the tree, it is usually closer to about a meter or lower, a meter from the ground or lower. And yeah, I do think that I see a lot in those like branching junctions in the lower branches.
Okay.
But we have seen attacks higher up in the tree, especially if like a higher branch has fire blight, then they can cue in pretty closely on like, here's where the stress is, and I'm gonna attack that same branch.
Okay, that's good to know.
We set up our traps though, our monitoring traps 1 meter, 1 to 1.5 meters off the ground. So that's like our typical sort of flight pattern is around that height.
Okay.
There is a comment that the beetles are tiny. And they're all ambrosia beetles are like 1 to 2 millimeters. Field ID is pretty difficult. I think you can tell it's an ambrosia beetle from looking at it on the sticky card, but to get a species identification is not something that is very easy. Especially between black stem borer and Anisandrus maisch, the differences between them are microscopic, and you have to take them off the card and look under a microscope. Which I'm happy to, if anyone has samples that they're interested in getting identified, you can send them to us and we can ID them for you. But I think in terms of identifying in the field, you can just say, yes, this is an ambrosia beetle, or no, it's not.
Well, for a grower perspective, is there any reason to need to go beyond just ambrosia beetle? Yes, no?
Depends on your situation and where you are. So I actually wanted to like go back to the spraying recommendations. I don't think you should be spraying unless you know you have attacks. Ambrosia beetles are going to come from the woodlot. The question is, are they attacking your trees or not? When it comes to ID in the field, again, if you know you have a history of attacks, you have reason to believe your trees are stressed, whether that's winter injury or you have a flooding type situation. I think that you're probably not going to be able to ID them on the card, but it is a way to just keep track of the history if you do find attacks too. Like you could send them to us if you then find attacks later, we could tell you what species you do have. But this is going to be highly variable from year to year. They're really dependent on temperature. So keep that in mind. Like some years it might take 2 weeks to escalate to peak flight from just seeing a couple. And then in other years, it could take 3, 4, sometimes even 5 weeks to escalate.
Okay, great. Thank you. And I think we're gonna hold you to that. It sounds like we can send you our sticky cards for you to ID them. I think we just heard it promised to your folks.
The sticky cards are sticky.
We do request if you do this, I cannot emphasize this enough. If you send us sticky cards, please wrap them in wax paper,
not
saran wrap.
not saran wrap. Not saran wrap.
They must have really hated you.
Yeah, and there's other, if you do use the bottle traps, there are other, they don't have to be the sticky cards.
The bottle traps are way easier to deal with for us. And we could send out. So didn't we write in a grant, Kelsey, that we were gonna do like a training?
Yes.
So if that gets funded,
TBD on if we get funding.
yeah, if we get funding.
But yeah, I think it would be beneficial at this point to put together some sort of handout in terms of Anisandrus maisch and black stem borer, if growers would be interested in that. Again, the ID without a microscope is not feasible for most people. But yeah, from the sticky card, you can say yes, Ambrosia beetle, no Ambrosia beetle. And we
Okay,
can
great.
ID if anyone wants to send us things. I'm gonna put my email in the chat if anyone
Yeah, please do.
wants to reach out. But yeah, we're still learning a lot about the second species, Anisandrus maisch, and how these two species interact in the orchards together. If there's going to be any sort of synergistic, if we see both these species in high numbers, is there going to be higher damages and higher loss? So we're still kind of teasing that out as we get more reports from growers and continue our own scouting and monitoring.
Mhm. Are there any other questions from the group? I see we're past 1 o'clock, so don't want to keep people too long, but certainly we've got time if there's anyone else that wants to ask anything or comment on anything today.
I would say if you're getting high beetle captures well into June and July of a small black beetle, it is highly likely that you do have the second species, Anisandrus maisch, because we do see a pretty steep drop off of black stem borer flight once we get into late June, early July. So just a heads up.
All right, and there's some discussion in the chat box there about people's preference for sticky cards versus the bottle traps.
Yeah, the sticky cards are quite goopy. I agree.
So yeah, I know we all have our preferences there, so. All right, well, not hearing other questions or comments, I think we'll go ahead and conclude for the day. I'd like to thank you, Kelsey, for joining us today. Really great presentation and great conversation in the chat. Monique, thank you for jumping in too, and everybody else that added in their comments today. So this was our last one of the 4 for this winter. So we have all the recordings available for the previous ones. We're going to get this one up in the next week or so, so keep an eye out for that. We'll send it out through the group email probably sometime next week. And then in the spring, look forward to starting up the weekly phone-in that Anna will probably, I assume, be leading up again this year. I don't know if we've talked about that yet, but I probably shouldn't assume. But hopefully
Yeah,
we'll be
it's a
checking
good
in.
question, Mike. I don't know if we'll be doing that or not. We'll probably be sending out a poll or something to gauge interest and the best timing. So look for that as well.
All right, thanks for jumping in there, Anna. All right, everybody. have a great rest of your winter. Hopefully a green tip doesn't come too soon for you all, and we'll see you next time.